have created a mitochondria diagram to show the outer membrane, the inner membrane, as well as the cristae, which you can see here. The mitochondria contains an inner membrane, outer membrane, and cristae shown in the diagram. This cristae also folds in the inner membrane of the mitochondria that allow the mitochondria space for chemical reaction to take place in. Oxidative phosphorylation takes place in the inner membrane of the mitochondria. There are 5 electron carriers, which are described here in my diagram. So as you see, we have 1, 2, 3, 4, and 5, which is also known as an ATP synthesis. And this is also shown as the far right. And our first one, the reduced coenzyme carries H plus electron to electron transport chain on inner mitochondrial membrane. And the second one, our electron pass from one electron carrier to the next in a series of redox reactions. The carrier is reduced when it receives the electrons and oxidized when it passes through them. So as you see, they pass through here. The next thing that happens is protons H plus move across the inner mitochondrial membrane, creating high H plus concentrations, and you can see that up here. H plus diffuse back into the mitochondrial matrix down the electrical chemical gradient. And then after that happens, the H plus diffusion allows AT pass to catalyst AT synthesis. Electrons and H plus ions combine to form hydrogen atoms, which then combine with oxygen to create water. If the supply of the oxygen stops, the electrons transport chain ATP synthesis also stops. As you can see in this diagram, if oxygen is not available to animal cells, then pyruvate is converted into lactate through pyruvate oxidation. Unlike animal plant cells, pyruvate is converted into carbon dioxide, which is not part of the eukaryotic oxidative phosphorylation process.